In this module, I will discuss about translation of mRNA in E. coli. What is translation? The process by which the genetic information stored in an mRNA is translated according to the specifications of the genetic code into the sequence of amino acids in the polypeptides is called translation. Steps in protein synthesis. First step in protein synthesis is activation of tRNA. That is, tRNA is activated by a specific enzyme. It is called amino acid tRNA synthetase. There are 20 amino acids there, and there are 20 specific amino acid tRNA synthetases are there for each amino acid. Now, activation of tRNA is completed in two steps. In step 1, amino acid reacts with adenosine triphosphate. The reaction is catalyzed by amino acid tRNA synthetase, which results in the formation of amino acid adenylate. In the step 2, amino acid adenylate plus enzyme, that is amino acid tRNA synthetase, reacts with tRNA, which results in the formation of amino acid tRNA plus adenosine monophosphate, and enzyme is released for further reactions. Stages of translation. There are 3 stages of translation: initiation, then elongation, and termination. Initiation factor is initiated by 3 initiation factors. They are termed as IF1, IF2, and IF3. Additionally, one molecule of guanosine triphosphate is present, plus initiator tRNA, then messenger RNA, and 30S subunit of ribosome. In the elongation process, there are 3 factors are there: elongation factor Tu, elongation factor Ts, and elongation factor G. One molecule of guanosine triphosphate and 70S ribosomal unit. Plus, there is a peptidyl transferase enzyme is required for peptide bond formation. Moreover, in the last stage, that is termination, there are 2 release factors are there: release factor 1 and release factor 2. In the initiation process, 70S ribosome, which is dissociated into 30S and 50S subunit. This dissociation process is carried out by the initiation factor 1. Now, this 30S subunit binds with initiation factor 3, which results in the formation of 30S plus IF3, that is initiation factor 3, which this initiation factor 3 prevents the reassociation of 30S subunit with 50S subunit. Now, in the 30S subunit, mRNA is attached, and this mRNA binds with 16S rRNA of 30S ribosome. Now, the binding takes place in the sequence. It is a purine rich sequence. It's a purine rich sequence AGGA-GG. This is a hexanucleotide purine-rich sequence which is located about 7 nucleotides from the initiation codon AUG. Now this sequence is called SD sequence or Shine-Dalgarno sequence after the name of its discoverer. Now this Shine-Dalgarno sequence paired complementary with complementary sequence of 16S rRNA, as a result of which initiation codon AUG is properly placed. When AUG initiation codon has been placed properly, the first tRNA, that is called FMET tRNA FMET, that is initiation codon AUG, code for the amino acid methionine. This methionine is formilated, so it is called FMET. Now this tRNA, that is FMET tRNA FMET, is brought in the initiation codon AUG by the initiation factor IF2. Thus, in the 30X initiation complex, IF3, IF1, IF2, mRNA and GTP binds. So this is called initiation complex. Now at the end of the formation of the initiation complex, 50S subunit then bind with the 30S subunit with the help of guanosine triphosphate, and IF3, IF1 and IF2 are released from the 30S subunit, as a result of which again there is a reformation of 70S ribosomal unit. As a result of the formation of 70S ribosomal unit, there is the completion of initiation process. After the completion of the initiation process, then elongation process starts. Now the chain initiation could be summarized as follows. Chain initiation begins with the dissociation of 70S ribosome and formation of two complexes. Initiation factor IF2 and FMET tRNA FMET complex is formed, and mRNA 30S ribosome IF3, IF1 and GTP form the initiation complex. Final step in the initiation process is the addition of 50S subunit to the 30S subunit to produce 70S ribosome and removal of IF1, IF2 and IF3 by the hydrolysis of guanosine triphosphate. Now during the elongation process, there are two sites are formed after the association of 50S with 30S subunit. One is called P site or peptidyl site, and another is called A site or amino acyl site. Now in the P site, the first mRNA which is added, that is methionine or FMET, it is present in the P site, and A site is remain vacant. In the A site, a amino acyl tRNA is entered with the help of elongation factor TU-GTP. So there are three elongation factors are there. One is EFTU-GTP, elongation factor EFTS and elongation factor EFG. So elongation factor EFTU-GTP is converted to elongation factor TU-GDP, which is inactive form. So elongation factor EFTU-GTP, it fill up the A site with a one amino acyl tRNA. Now EFTS, which is another elongation factor, which is again activated EFTU GTP into EFTU GTP. In the next step, that is peptide bond formation step. Now from the P side, this FMAT, it is transferred to the A side and a peptide bond is formed. A peptide bond is formed by an enzyme. It is called peptidyl transferase enzyme. And tRNA, which is emptied, which is ejected. It has also been suggested that there is also an additional E site is there. Now, in the next step, that is from the A site, this nascent amino, that is nascent polypeptide, is moved to the P site. Now, this process is called translocation process. Translocation process is catalyzed by another elongation factor, that is elongation factor G along with guanosine triphosphate. During translocation process, that is ribosome, this ribosome, it moves on the mRNA to the left side, as a result of which a new codon is entered in the A site. A new codon is entered, as a result of which a new amino acyl tRNA is inserted in the A site. Again, from the P site, the nascent polypeptide is moved to the A site. Now, this process now continue and tRNA, which is free of amino acid, is ejected. So, polypeptide is being formed in this elongation process. We can summarize this chain elongation process as follows. In the first step, that is binding of an amino acid tRNA to the A site of the 70S ribosome, transfer of growing polypeptide chain from the tRNA in P site to the tRNA in A site, and formation of peptide bond by peptide transferase. In the third step, translocation of the ribosome along the mRNA to the position of the next codon in the A site. Additionally, this peptide transferase step in prokaryotes is inhibited by an important antibiotic. It is called chloramphenicol, because this chloramphenicol, it mimics the amino acyl tRNA and it inhibited the protein synthesis process. The last stage of the protein synthesis is termination. Now, this termination process is initiated by the termination codon, that is UAA and UAG, which is recognized by release factor 1, and UAA and UGE, these two termination factor is recognized by release factor 2. Now, when a termination codon is present in the A site, so release factor will enter into the termination codon, as a result of which the elongation process stops and nascent polypeptide is released from your A site. Subsequently, after the release of the nascent polypeptide, the ribosome, 70S ribosomal unit is dissociated into 30S and 50S subunit, and mRNA is also detached from this ribosome, as a result of which the termination process is completed, and again the protein synthesis, that is initiation process, will be started. So we can summarize this chain termination as follows. Polypeptide chain elongation undergoes termination when any three termination codons, that is UAA, UAG and UGA, enters the A site on the ribosome. Stop codons are recognized by release factor 1 and release factor 2. Release factors adds water molecule to COOH terminus, which releases nascent polypeptide. Termination is completed by the release of mRNA molecule from the ribosome and dissociation of ribosome. Ribosomal subunits are ready for another round of protein synthesis. In this module, we have provided key concepts of the module. Some quizzes are there, list of books for reading and study materials. Thank you for attending our online course.